slide. We were talking about how through time, if environments change, or if there is a selective pressure, a benefit or a detriment, we can see changes in the gene pool because our gene pool is not stagnant. Our gene pool is extremely malleable. It can change very easily. And we talked about how we can see that ourselves looking at artificial selection we show the picture of the dogs. And I'm not gonna go all the way back, I don't know where it's at. But the dogs, and then we show the picture of now I messed it up. And if you think about the wolf, the genetic ancestor of the wolf, over the past 100,000 so years to 10,000 so years ago, we were able to, well, there are many hypotheses, either they domesticated themselves by hanging out with us, or we selected and bred for the more docile, the more tame. But then over the centuries, we selected for smaller dogs, cuter dogs, bigger dogs, dogs that could hunt, dogs that could dig, dogs that could follow us around, dogs that could pull sleds, all kinds of stuff in order for us to change that genome and to see the phenotype that we want to see. We did the same thing with all of our food, all of the different types of vegetables that we eat. That's not what they look like in the wild. That wasn't what their ancestors looked like. We just took those and we bred them and bred them and bred them until we saw what we wanted to see, until we got the product that we wanted to see. That's artificial selection. Natural selection is the same thing, only we don't have a hand in it. It's just what happens out in nature. Either you're living or you're dying based on selective pressures. And either you can have some sort of beneficial adaptation and survive out there in the world or not. Changes don't have to happen. There's no rule saying you have to change. You're only changing if there is a selective pressure against you. And so we talked about like the alligator and crocodile lineages. That group has been around forever, before the dinosaurs. Or horseshoe crabs, those little tiny, long, flat crab-like organisms that hang out on the bottom of the ocean seafloor. That lineage has been around before trees were in existence. And so you don't have to change. But if there is a changing environment, if there is a selective pressure against you, over time we are going to see changes in those genomes. We're limited by, right now, by our human brains, right? We can't go back in time. And we only have, so many years that we've been writing things down and keeping track of stuff. And so the only way that we can understand what once was is to look at traces of the past. So we can look at fossil evidence. We can look at biogeography, where things live and where they've moved to, where ancestors used to live. We can look at biochemical evidence as well. Because the DNA, the genome, that's a map of what has changed, and we can trace that back. And so, we're not quite at the science fiction phase yet where we can just take a bit of DNA and then, have an image of that organism, but we can map those differences. And so we were moving into fossils and talking about how fossils are any trace of the past. It doesn't have to be a bone. It could be, a footprint. It could be droppings. It could be something left over, a leaf, amber, anything. A fossil is just a record of what once was. Now, we'll talk about fossils a little more in lab this week, but let's look at this image here. When we're trying to understand ancestry, it would be absolutely impossible for us to make a map of every species that has ever existed. And so when you see these types of images, when we show you what we'll eventually learn as a phylogenetic tree, what we do in these pictures is point out key features and then use these kind of maps, these family trees, to show you how those key features have changed over time. This isn't listing every single species. This isn't saying this became this, became this. We're just picking out the key features of that family tree. And the whales, I think I told you, one of the last things I said on Friday, are some of the most amazing organisms on the planet, which I didn't even think about till later in life. But I mean, the blue whale is the biggest thing that has ever existed on this earth. Isn't that wild? And it's huge, and it lives in the ocean. It's just, and it filter feeds basically. And so it's insane. But the whales are mammals. They have terrestrial breathing systems, which means they have to breathe atmospheric air. Their live young have to breathe to jumpstart that circulatory system as soon as they're born. They produce live young. They produce milk. And so they're not exactly, some of their leftovers aren't exactly the best for a life underwater. But what has happened is these traits have changed. We've seen this malleability in the genome. And so millennia ago, whales kind of came off of lineages that were more seal-like, they think amphibious perhaps. And there were these ideas that, okay, if this was true, we need to see some of these key features change. Now, a big one with your aquatic organisms, your aquatic mammals, is they have different ear bones than we do. Because we have ears that are adapted to hearing air or a sound through air. And so we have to pick up to that delicate little membrane we have on the inside. If you go underwater too deep, that's going to burst. That's a terrible thing. And so they have different ear structures than we do. And so you can map these certain changes in their ancestry. And so we have just 3 organisms here: Ambulocetus, Basilosaurus, and then a modern day whale. There are changes that you can follow through these lineages. A big one being where they breathe, how their nose essentially move back over time. Interestingly, the hind limbs, once you move underwater, you don't really need to walk anymore, especially if your lineage, your family line isn't even going back to the mainland. And so through time, we see how features that are already there can be shaped into what you need, essentially. Because you have to get rid of a misconception. Maybe you don't have it, but maybe you do. Some people have a misconception that evolution or natural selective pressures choose for a magical trait, and that just gets popped out of the ground. Or, you go to sleep one night, you wake up with gills, or, oh, these whales needed fins. Next thing you know, they have fins. You can only change what's already there. I mean, you only have, your genome is your genome. And so what we saw through time was a feature that they already had, which was the hip bone, walking leg bones, just become morphed or changed into eventually a feature that can be used for something else. Because that's what life does. It takes features that you already have and then use it for something else. Because it's a lot easier to do that than it is to just create a brand new feature. But we'll talk a little bit more about fossils in lab. Let's move on to the next. Are there any questions about fossils so far? No? Good. All right. Biogeography is the study of where these species are and where they exist. And so this played a big role in understanding how organisms have moved through time and how those changes have developed. And so throughout the Darwin's voyages and then Wallace, when they were collecting fossils and collecting drawing and collecting samples of living organisms, they were able to compare what once was to what is now and where things exist. So if you think back to one of the things we said about Darwin, we said natural selection, if it is occurring, should be changing organisms to fit that environment. We should see organisms adapted to that environment. It wouldn't make any sense to see those that didn't fit in that environment because they wouldn't have lasted. And so we can kind of look at all the different areas on the planet and look at the different organisms and see what they look like and see how they have changed through time. Australia is a great example of how when organisms are separated over a large amount of time, but exist in similar habitats, we will see similar features. Because Australia was ripped apart from the main continent a long time ago. So those organisms were on their own for a long, long, long time. But we see very similar organisms. And so if there's a similar adaptive pressure, we're going to see similar features. But we know Australian mammals are extremely different than a lot of our mammals because they're marsupials. So rather than have the placental mammals that produce live young ready to go after birth, they have to hang out inside that pouch. They have to carry their live young. They live inside that marsupial pouch for a while, which is completely different than some of the mammals that we saw today, because that was the lines that they evolved from. But because there were similar habitats, we see similar organisms. And so marsupials evolved from egg-laying mammal ancestors, and you still see that in some things. You can see that leftover. Do you know what a platypus is? Have you seen those before? They're kind of mammal-like. They're a very primitive image, so they're kind of like a proto-mammal. We call them monotremes. They still lay eggs. They never developed that marsupial pouch or a placenta. And so there that lineage branched off of that. And so there was no rule saying everything has to become a live bearing, mammal or a mammal that bears live young. It was just a leftover and it fit. And so looking at these different organisms across the planet allow us to kind of look at how features have developed. And so an organism that went extinct in the 1900s, the Tasmanian wolf, shows extreme similarities to other canines, or thylacine isn't a canine, but to canines that, like the wolf, dire wolves, things like that, but are distinctly different, but they were just in similar habitats, occupying similar roles, playing that predator role at the top of that food chain, and so we saw similar adaptations develop. The sugar glider is very similar to our flying squirrels that we have, similar organisms in similar habitats, but not closely related. And so biogeography allows you to compare that and see where they branched off from and where they exist now. Another good example of looking at biogeography and changes over time is the camel lineage. Camels are an older group of mammals, developed ages and ages ago in North America, but as those organisms migrated, you see changes develop over time. So if you think about the South American llama, the Bactrian camel of Africa, the dromedary camel of Asia, these organisms look pretty similar because they're distantly related. They develop from the same family line. Now, they're not the same species at this point. They can't interbreed necessarily, especially the llama. They live in different areas, and so through time, they were adapted to that environment in which they live. But they all arise from a similar ancestor, a similar ancestry. Same thing with the giant salamanders in Asia and the hellbenders that we have in our streams. Obviously have not been geographically close in a very long time. North America and Asia are extremely far away, but those organisms share common body plans, common habitat adaptations, similar roles in that environment because they share that ancestry. And so biogeography played a key role in understanding not just natural selection, but understanding time, geographic time too. But because remember, in the 1800s, the common idea was Earth was only 6,000 to 8,000 years old, maybe 10,000 years. But the idea of 100,000 years, a million years, a billion years, that was insane to people. And so heretical, in fact. And so it was these concepts, looking at the plants, looking at the rocks, looking at fossil remnants across the planet, we're able to compare and see that the Earth has changed quite a bit, and it is pretty old. And so we were able to develop the idea of plate tectonics, the continents moving around, and organisms migrating over time. Things that we've been hearing our whole lives, and so it seems kind of common knowledge to us, but they didn't have the type of, you sometimes need to put yourself in their shoes and think it's not that they didn't understand, or they weren't unintelligent, or they were, we're all resistant to change, but it was more they just didn't have the way to collect the evidence like we do. We can measure the movement of the continents these days, and it just wasn't something that could be done. When you're looking at fossils, we're going to look at two types of features. If we're looking at fossils to compare ancestry and to compare traits, we look at two types of structures. One we call homologous, which means you have it because that's what your ancestors had. Maybe it doesn't do the same thing anymore, but it's left over because that's what was in your family. I call it family line, but your ancestry. It's not really a family line. It's just when I picture this type of stuff, I picture like a family tree, but just on a grand scheme. And so there are also analogous structures, which means structures that look alike or do a similar thing, but aren't due to common ancestry. It's due to that selective pressure, molding you to that. And so homologous structures as examples, if you look at all the mammals, we have a ton of homologous structures. Maybe they don't do the same thing, but we have similar body plans, similar bone structures. So as vertebrates evolved, bones are put to different use, but we all still have that same plan. And so if you look at the skeleton of organisms, you can see that. You can see these homologous structures, things that maybe do different things now, but the bone plans are the same. And so look at these organisms here. Here's us. You'll see this in lab. They've sort of taken and colorized humerus, radius, ulna, the wrist bones, the carpals, and the finger bones, the phalanges. Other organisms, other vertebrates, have that same body plan, but through time, they've been adapted to various scenarios. So for example, look at the bat. Look how long those finger bones are now. And so those similar bones were still there because you can only modify what's already there, what's already in your genome. You know, maybe it wasn't the most beneficial to take the hands and turn into wings, but it's not like they had a choice in the matter. It was just what their genome was. Same thing with a horse. Horses are essentially walking on what we would think of as a finger. And so, but it works, right? Horses came about when the grasslands popped up after the last big ice age, and it works really well. Not the best, because natural selection doesn't always choose the best feature, just what's going to keep you alive. And so anybody know what the Kentucky Derby is? You know, we watch horses race all the time. You drink your mojitos and things, fancy big hats. With horse racing, horses, they break their arms, essentially, their phalanges all the time. Because they run, essentially, they develop that adaptation to escape the predator off the grassland. So you'd run, they have a lot of endurance, they can run for a long time and outrun predators. They can only run so long, so that structure isn't the strongest. We'll see cow bones in lab, we'll have a horse bone and a cow bone, and we'll compare and see just how thick the bones of the cow are, because cows don't necessarily walk on their, you know, what we call fingers like horses do. But these adaptations aren't perfect. They're just what occurred based on what genome you have. And so these homologous structures develop over time based on what you already have. And so birds and dinosaurs, which are part of the same group, a separate lineage from things like crocodiles and pterodactyls and pterosaurs, what we call non-avian dinosaurs. So the avian and the non-avian all have similar structures. Similar genome patterns, just put to different use. And so the large dinosaurs, the big ones that we see, like when we think Jurassic Park style, aren't around anymore. They all died out after the last big extinction movement after the Cretaceous. But their family lines are still here. The birds, essentially, all the what we call birds, evolved from what we think of as dinosaurs. But when we start seeing these organisms and start understanding what they looked like and how they acted, and how they put those features, the difference between the two becomes smaller and smaller and smaller. We begin to understand, well, of course they're similar. I mean, you ever seen the way some of those birds go after food, the way they hunt, the way they look? They seem very raptorial, very, you know, stereotypical dinosaur-like. And so sometimes when we try to categorize things, once we start to understand, it makes much more sense. Especially with the birds and the dinosaur grouping. They've done they had to use RNA testing, but they have sort of like a fragment of a genome from a Tyrannosaurus fossils, and they compare genomes to all the living organisms. Have you heard this story before? Do you know what the closest living relative to a Tyrannosaurus rex is? No. It's the chicken, the modern day chicken. So can you imagine Tyrannosaurus rex is like, well, this is your family line now, like not exactly the strongest, but they lay eggs. So just over time, you know, these features, just you lose them, you gain them. The biggest like aha moment with this was when we uncovered what we now call Archaeopteryx, and especially all of the microraptors, which we're finding in China and Asia. These organisms, these fossils were so well preserved that we can basically see not just the bones, but the leftover of what they had around them. And so we kind of get the Steven Spielberg Jurassic Park type image in our head of just like this reptilian skin. But most avian dinosaurs had feathers. Feathers developed as a display, probably, and as thermoregulation, probably, before flight. But then they allowed flight, the adaptation, to develop. And so we see these organisms, these smaller dinosaurs, with feather patterns And then Archaeopteryx was the first one we ever saw that had bird-hipped or lizard-hipped and gliding feathers. And so we now find most of these types of organisms, the avian lineage probably did have feathers. Your things like Brontosaurus and those Stegosaurus, those things probably did not, but the more avian type did. And so these homologous structures are structures that allow us to see ancestry. They're there because that's what the ancestor had. And so if you're looking at dinosaurs and birds, they both had hollow bones, because if you have these large bones, you have to carry that around, your bones are hollow. Now that they're hollow, they're lighter. So when you're gliding, they're not weighing you down. The birds and avian dinosaurs have the same skull structure, same hip structure, same breastbone structure. Their hearts are similar. They have the branching off of their aorta than other dinosaurs had. They have what's called gastroliths, which means inside of a crop, when they eat, if you ever had chickens or raised chickens, they have to get gravel inside of their food or some type of rock to help crush that up before they can digest it. Dinosaurs, the avian dinosaurs, had the same digestive system. Sleeping patterns they recognized were probably the same. They can tell this based on where things have, where the brooding were, where they find nests, where they find these locations. And so they had similar features. And so now they're so similar that we know that they're not even a separate group. Now, analogous, this is when people miss a lot. They get confused with it. An analogous structure is a structure that exists, but isn't necessarily due to common ancestry. It just does the same thing. And so it may not be constructed similarly. It just does the same feature. And so one of the easiest ones to look at are body plan and then flight. Sometimes if you have organisms in similar habitats, they develop similar features. We've been talking about that. That's because the pressures are the same. And so we had Darwin's Patagonian cavy and the European hare. Similar environments, different parts of the planet, they looked the same. Why was that? The similar pressures. This is called convergent evolution. It means the pressures are the same, so you're going to see similar looking organisms. So think about all the things you can think about. I should have worded that differently. Think about everything you can that lives in the ocean. What sort of pressure do you have in the ocean? It's big, it's broad. You have to look out for predators, have to move quickly, have to be able to withstand the pressure of the water, have to be able to find food. And so if you think about organisms that exist in the ocean as their main form of survival, you see similar body plans. You think about the shark, which is a fish, dolphin, which is a mammal, penguin, which is a bird, ichthyosaur, which was the dinosaur. You think about seals, think about manatees. What body plan do they have? But that similar sleek body plan, oftentimes fins to help for turning, similar features. That's because the pressure is the same. So in that environment, the pressure is the same. You're going to see similar body plans. It doesn't mean sharks and dolphins are closely related. Dolphins are a mammal. That's an extreme difference. Sharks are a fish. They're a very, very old lineage. So they're not closely related at all. Sharks have cartilage, dolphins have bones. And so there's a difference there, but the body plan is the same because the pressure is the same, the natural selective pressure. Now, an analogous structure could also be like flight. And so we just said that the avian birds and pterosaurs are two different lineages, but they both fly, right? They have similar features. If you think about your flying insects, they can both put a structure to that use for flight, but they don't have recent common ancestry. I mean, think about a butterfly and in this picture, a duck, right? They're not very closely related at all, if you could imagine, but they still have that flight structure. That's an analogous structure. Homologous is you have it because your ancestors had it. Maybe it does something different. Analogous is it does the same thing, but not due to common ancestry, just due to natural selective pressures. You okay with that? We'll see it again in lab. You'll have another time with this, so you'll have a chance to develop that further. We don't get too much into embryology in here because that needs us to understand a little bit more about development in the cell than we get in Gen Bio. We get a little bit. Embryologically, if you think about an embryo inside of an egg, inside of a placenta, what sort of pressures do you have as an embryo? How did you have to survive as an embryo? You had to get oxygen, you had to stay warm, and you had to get nutrients. That's about it. And so there aren't many selective pressures against embryos. And so what we see embryonically with organisms, especially if they're in the same group, we don't see a lot of differences in the embryos until other features start developing. Because when you're first developing, you just got to make sure to get oxygen, nutrients, and stay warm. That's about it. And so natural selective pressures haven't changed many embryonic structures. And so we see a lot of leftover. And so invertebrates, as an example, if you look at vertebrate embryos, if you didn't know what you were looking at and you didn't have access to the genome, our embryos look very, very similar because we have that backbone that has to develop, a brain that has to develop, and then our structures start developing as we age into what our lineages are, into what our group is as the cells start recognizing who they are. But in that first plan, we're very, very similar, a lot of similar structures. And interestingly, as humans develop, we have two leftover structures that we don't lose for a long time. One of those is a postnatal tail. Obviously, we don't need tails anymore. We don't have to balance. We don't need those. And so our vertebrae is smaller and tucked in, unlike our other groups. And we have what's called pharyngeal pouches. These little pouches on the neck that develop that obviously we don't have to have anymore. And so these are just leftover of where gill structures used to be once upon a time, but vertebrate lineages don't need those anymore. And so you lose them as you develop. Going back on that slide at the bottom there, you can also see leftovers in your history, which are some of the coolest things. They call them vestigial, which means they don't do what they used to do. So they could not have a use anymore at all, or they could just have sort of a benign use that doesn't really do much anymore. If you think about it like our fingernails, I mean, we don't really have to use those anymore, but they're still there. Our wisdom teeth, we don't really need our wisdom teeth anymore. Our jaws are much smaller. We have dental care these days, but some of us still have them. But because our jaws are much smaller, sometimes they come in impacted sideways. You have to get them removed. They get abscessed. They're just a leftover. And they have a reduced or obsolete function or a function put to other use. And so these vestigial structures, you can see them. One of my favorite are looking at the flightless beetles. We have some beetles that hang out all the time in this desert environments on the ground. They burrow under the sand, and they have this hard, super hard outer shell. So beetles have two sets of wings, an outer wing and the inner wings, which are their flight wings. In these flightless beetles, their outer wing has fused completely. They don't use it anymore because they have a terrestrial underground type fossorial lifestyle. But if you were to dissect those outer wings off, they still have the flight wings underneath because that's what their family line had. They don't use them anymore, but they're still there. Do you know what a manatee is? Or a dugong? Sort of like big, slow sea cow things. They've been sort of aquatic for a while now, but they branched off from the same group where we would find like hippos, cows, whales, that sort of group. They branched off of that. Elephants is a big one. And so if you were to look at their flippers, if you ever see one up close, take a look at them. Some of them still have leftover giant fingernails, which look like, you took an elephant's foot and just squashed it. Well, obviously these things aren't using those structures anymore, but there's no rule. Nobody's saying, oh, we gotta get rid of that. It's just a leftover of what once was. So blind mole rats, if you dissected the flap off their eyes, they still have leftover eye structures. They don't attach anymore, but they still have the genome to make the leftover. It's just not used. Some self-pollinating plants still have, ovaries and carpels that they don't put to use anymore. And so sometimes we can see the leftovers because there's no rule saying you have to change, no rule saying get rid of that, keep this. It's just what you pass on to your offspring. Now, I wish we had more time for this section and we don't, so I'm sorry. If you ever take genetics or something, you'll get more of this. Our genome sometimes structures reemerge because we only have so much in our toolkit and what our genome is. And sometimes natural selection or random events can cause lost structures to reappear, and we can use them again if we have to, or there's just a mutation that pops up because of that plasticity. And so we learned, if you think back to chapter 12 and 11, that some genes can be turned off and some turned on. We have a lot of leftover in our DNA of genes that are just turned off, and we don't have the ability to turn them on. But we're kind of advancing in our science ability, and we're getting to kind of the science fiction age with our technology. We know now how to turn some genes on. And so we see some of these genes pop back up occasionally. Whales don't have their hind fins anymore, but occasionally they're born with them. If you think about dew claws on dogs, not all dogs have them, some dogs do. Sometimes things that we thought were flightless, some insects can still be hatched with those wings because it's just a leftover. Did I take it out? I took it out. Let's see if my link still works. This is a couple years old. I like how I took it out and I still talk about it. So there's this link here you can read if you would like. I was talking about how we can turn genes on and turn genes off. They probably removed the picture. They did. We said that, or if you remember, I said that birds are part of that avian dinosaur group. Think about a bird's mouth. Most of them don't have teeth like we think of, like dinosaurs do. But those genes for teeth are still there. So what we did is a group took chickens and turned the genes for teeth back on. And those embryos developed with teeth. They looked just like a little tiny dinosaur almost with their little teeth structures. They didn't allow them to live because it gets into a weird ethical thing of should we turn genes on or off, and should we be allowed to do that? And so they didn't allow them to live, but those genes are still there. They're just not put to use anymore. And so sometimes we can see an emergence in those lost traits, and a lot of times that's how we see major adaptive change across the fossil record, is sometimes genes got turned back on. My watch thinks that I'm exercising. We can also look at our DNA. We can look back and sort of measure what we already had. So all living organisms share the same basic molecules. They use that same triplet code. And so we use the same 20 amino acids. We learned that. Now we all have it in different patterns, and we can look at that pattern. And the closer your pattern is, the closer you are genetically. And odds are the closer you'll look. And so evolution can just be a modification or duplication of what you already have most of the time. Now, some of these last parts here, I'm going to skip through pretty quickly, but I'm going to point out the important parts. And so get it out of your mind that evolution is just coming up with a brand new feature. Oftentimes it's just either turning a feature off or duplicating a feature you already have. And so you can't just come up with a brand new trait. Oftentimes it's just what you already have. And so organisms that have, a lot of organisms have the same exact body plan. We have a toolkit, our genes that code for where the head goes, where the arms go, where the legs go. But we just have a modification in that genome. And so as an example, organisms like, if we look at like a snake, mouse, human, we have the same body plan and genes that say, hey, your head goes here, your neck goes here, your vertebrae go here. But the more vertebrae you have, the more of those genes that you have. And so we were just able to take our genome and duplicate it. And so from our genomes, we can tell who's closely related and who's not. Do you remember what the mitochondria are? Yeah, they can make ATP, make energy, right? Those have been in our eukaryotic cells for a long time, long time. We're talking back at the cell level, the cellular level, unicellular level. And so all of us eukaryotes carry around mitochondrial ancestry. There's a very particular protein inside of the electron transport chain called cytochromes. And one called cytochrome C has been kept around for a very long time. And if you look at the mutations and the differences built up in the cytochrome C complexes, the closer those mutations are, the closer that cytochrome C protein is, the closer you're related genetically. The further away that is, the more distant you are. And so the cytochrome C, if you take cytochrome C differences and similarities and sort of lay it or map it over the hypothesis of our big family tree, it all lines up. It sort of reinforces the idea of how we have branched off from other organisms and reinforces who the closely related organisms are and who they are not. And so this image kind of gives you an idea. Now, it's not saying that moth became fish, became turtle, became duck. I get that sometimes, and that's not what this is showing. It's just listing common organisms that you probably have heard about, and then showing you that genetically, there are some similarities between groups. So between primates, between mammals, between vertebrates, between all reptiles, between all fish, and the insects branched off a long, long time ago. In the cytochrome C complexes, when you look at these, you have to read your tables. You can't just look at the top. It's just sort of saying that the more amino acid differences we have in that protein, the further related we are, and vice versa. I have a couple minutes left. I will have to finish some of this on Wednesday, but the last half of this, a lot of it is just sort of stuff that I'll point out just the vocab words, the stuff that will be on the exam. We were talking about how when we see similar selected or similar adaptations, but not due to common ancestry, it could be because they have similar pressures. And we call this convergent evolution. The pressures were the same, so the features look the same. And so as an example, sharks and dolphins. So here are some pictures of organisms that have raptorial forearms that allow them to grab a prey and then eat it. These structures have developed multiple times. These organisms look alike, but they're not closely related. And so praying mantises evolved in the Cretaceous, mantis strip evolved way back in the Permian, but they still have that similar feature because that raptorial arm allows them to act in that predator role. So even though these features evolved multiple different times, we see a similar adaptation because of a similar pressure. And so that's just called convergent evolution. The pressure was the same, so the feature we see is the same. And so there are some misconceptions, and I do want you to look at that paragraph in chapter 15, but not everything will form a fossil. Not every fossil will last forever. We have a much broader understanding of fossils than they did in the 1800s. And we're finding stuff, it seems, every year, new things as we get better at looking. But not everything forms a fossil. You have to be in a very particular situation. It has to be kept away from the elements, protected, so underwater, under a mudslide, out of the way of UV light that will break it down. So not everything will form a fossil. And so we don't have fossils for absolutely everything, but we have a ton. And so these are hypotheses, multiple hypotheses, that we can lay out to show these plans of changes over time. But obviously we don't have a fossil for every single organism that has ever lived because fossils break down. But over the span, we have quite a bit. And chance does play a role, but evolution and adaptation isn't necessarily random because you have to have selective pressures. It seems like occasionally the feature will be random, whether or not a trait is turned on or turned off, or you have a little bit of a different selective advantage, but the survivability isn't random because either you survived and made babies or you didn't. And so things don't just happen all at one time. Complexity doesn't just happen because it can only be a modification of the genes that you already have. And I think that's a big misconception that people carry, especially depending on how you have learned this in your past and who your instructors were. And so sometimes you learn these features that, things go to sleep and they woke up with gills. Next thing you know, we had turtles, and next thing you know, we had birds. It doesn't happen like that. It's only a modification of what's already there. And so a change of what you already have. And so evolution and natural selective pressures act on the whole individual, not just one trait at a time either. And so through this, we can observe and we can test these changes. And so with natural selection and with evolution, these are no longer hypotheses, but the unifying concepts that hold biology together, essentially, that sort of lay the foundation for everything, and that bring in the other sciences as well, bring in geology and chemistry and other types of physics. It all just sort of gets connected by this concept. And so we will look at that video. There are a lot of words here, but let me point out the big stuff. And so sometimes, you know, because natural selection can only act on what's already there, there are some limitations. And so selection can only remove phenotypes. Natural selection can only remove phenotypes. Mutation adds new things. Selective pressure always select against, not necessarily always for. That's why you have these leftovers and the imperfect adaptations, because it's not always what's going to make you do better, but what's going to make you do not as bad as everybody else. Does that make sense? And so we get these imperfect adaptations. And so I'll make sure to do some review on Wednesday, but let me just move to this. It'll be the last thing I talk about these last 4 minutes. With imperfect adaptations, you look at organisms and you say, well, that's kind of weird. I mean, look at our lungs, right? We have to breathe constantly. We can't get them wet, right? We can't hold our breath for very long. We can't just take one big gulp of air and be good. But it's obviously allowing us to live a lot better than those things that aren't. And so it's imperfect adaptation, but it does just fine. Look at whales. I talked about whales over and over and over again, one, because they're super cool, but a marine environment, but you have your live young, and if you don't have your baby near the surface of the ocean, near the atmosphere, and it doesn't get that first breath of air, it's gonna die and drown. Isn't that crazy? But it works, and it works okay. But the babirusa, you can see those at the zoo in St. Louis. They have these tusks they use for mating, and throughout their reproductive cycle, they can mate well enough to survive, but eventually those tusks grow into their skull and kill them. But throughout the time that they're reproductively fit, they do well enough to pass on babies. And so it just works, even though it's kind of imperfect. And so some species, we see these imperfect traits, or we see leftovers. Slugs and snails, those groups have gained and lost their shells over and over again. And we have organisms that exist kind of in the in-between right now, but it doesn't hurt anything. It's not hurting their fitness. And so it works just fine. And so we see these leftovers and these imperfect adaptations. Because there is no goal, it's just what makes you not do as bad as other things. And so digest that. We'll finish up just a little bit of this. We may not finish this whole PowerPoint. And then Wednesday, we will move into chapter 44.